All right, we're live. Hello, everyone. It's week 12. It's me and Kelton this week. We're just going to go through what we got for our answers. This is number 1. Which of the terms describes or is an example of a regulatory protein? That would be your cap, activator, and your repressor. Which of the terms describes or is an example of a small effector molecule? That would be camp, allocase, inhibitor, and inducer. And then which of the terms describes or is an example of a sequence found within the DNA? That's your +1 sequence, -10 sequence, lock Z sequence, and your lock 4 sequence. Which of the above is not involved in the lac operation? That's your inhibitor. Which of the above is an example of a structural zine? That's lock Z, and lock A. And for our fill in the blanks.
So fill in the blanks is pretty simple. Just read it across and fill it in. We got these from our notes in the PowerPoint. So we just filled in the lock operation is regulated by 2 inducers, the camp and the allocase or alloctase. And then B is the binding site for sigma factor and RNA polymerase is called the promoter, while the cap site is the binding site for the activator that regulates the lac aperon. And then C, the enzyme andinyl silase converts ATP into the C-amp. And then D, the -35, -10, and the +1 sequences are found within the lac promoter portion of the lac aperon. E is the lac O gene, encodes the lac repressor, while the lac Y gene encodes lactose polymerase or permase. F is attenuation can regulate gene expression in bacteria by causing transcription to terminate prematurely. And then G, an E. coli strain that contains 2 copies of the lac aperon is called a mutant strain.
For number 3, we went through and just figured out how to decide if the B-gallicidase and if it was going to be part of the where's it at? If that'll result in blue E. coli. Sorry. So when we have glucose absent, but then we have the IPTG present and the X-gal present, that's when you'll have blue E. coli. But all the other ones would be no. So, and then in 4, we just used the little key that she gave us right here, and that helped us to decide if it was constructive, inducible, or non-inducible. So for A, we have inducible. For B, we have non-inducible. For C, we have constructive. And D, we have non-inducible. And E, we have constructive. And then the same thing kind of goes for on 5. Me and Kelton kind of did this one a little bit separate, but got pretty similar answers. Using what we found in number 4, along with her little sheet that she gives us right here. You can figure out what type of yellow it'll have, whether it's 0 yellow, 1 yellow, or 2 yellow. So what I got for A was 0, 0, B is 1 and 1, C is 2 and 1, D is 0, 0, E is 0, 1. And I think on Kelton's he had D as 1 and 0, and then E was 0, 0. So we just had D and E swapped. But I'm just going to keep mine how it is because I don't know who's right or who's wrong. But that's what we had for the week.
Works good.